Thank you very much, James, for agreeing to give a Build a Cell seminar, and you can get started.
Great. Can people hear and see me okay? Yeah.
Yes.
Okay, great. All right. Well, thank you so much for all attending. And thank you so much, Kate, and the Synthetic Cell Group for this invitation to talk today. I've been on the edges of kind of synthetic cells, and so it's really great to kind of come and share the work that I think, although I'll present it for a slightly different application, I think, as you'll see, is very related and hopefully applicable to the stuff that you all care deeply about. So my name is James Chappell. I'm from Rice University. Quick overview of kind of where I'm coming from and my prior work. So I received my PhD from Imperial College in the lab of Paul Fremont. My interest here was really on cell-free systems, which of course people work with a lot in synthetic cell community. And we were really interested kind of early on in some of the early applications of these technologies. So particularly for prototyping of genetic parts and circuits, as well as building biosensors. After this, I went to the laboratory of Julius Lux, first at Cornell, then at Northwestern, where my interest shifted a little bit more towards component engineering, and particularly thinking about how we could engineer synthetic RNAs to create regulators of gene expression and genetic circuits. After this, I joined the faculty at Rice University in 2017, where my lab has kind of shifted directions, and I'll get into that now. So, when I started the lab back in 2017, it was a really interesting kind of time for the field of synthetic biology. I think we're starting to see deliverables of our ability to be able to engineer often quite complex phenotypes and functions in microbes like E. coli, right? So at this time, we could really engineer robust dynamic behaviors, so things like oscillations. We could engineer, quite complex computations into cells, and of course, biochemical transformations. Now, while we were able to do this in our, model microbes like E. coli, one limitation I felt at the time of the field that was holding it back was that most of these successes had been in a handful of model microbes, so most famously E. coli, and mostly under very artificial conditions of kind of pure culture and laboratory environments. And for me, this is kind of limited in two ways. Firstly, focusing on just E. coli or a couple of model microbes means we're ignoring the massive diversity of microbes that have evolved on this planet. And so I was really interested in trying to push into more non-model species. And of course, it's limiting because, microbes don't grow in pure culture in the lab, right? They, of course, naturally grow in microbiomes, right? These kind of complex communities, they're full of non-model species, often residing in quite complex environments and matrices. And I just have some examples shown on the slides here. So as a lab, I got really interested in trying to push the kind of genetic engineering frameworks to be able to deliver on this type of genetic programming of non-model microbes and particularly microbiomes. And the approach that we decided to take, influenced by my training, was really to kind of leverage RNA-based technologies. And I thought that this was going to be a successful approach for several reasons, and why I thought RNA was a particularly powerful molecule to do this with. Firstly, RNA is a very versatile molecule, right? So, while for a long time we thought of it just as this kind of passive intermediate as you go from DNA to proteins, we have since realized it does a lot of things, right? It can sense, it can catalyze reactions, it can regulate gene expression. And so as a molecule, right, if you invest in understanding how it works, we can do lots of things with it. RNA is also very designable. I think this is less important in the era of kind of de novo designed proteins, but at the time, RNA was one of the few molecules where we really had a great shot at trying to understand deeply the relationship between sequence, structure, and function, right? And I still think RNA is a very designable molecule, and there's kind of benefits to it. Finally, for our particular applications that we cared about, one hypothesis of the lab, which I think we have since proven is true, was that a lot of these RNA systems were going to be highly portable across diverse microbes, right? And so they would be easily be transported from our model microbes like E. coli into more exotic bacteria. Okay, so when we started the lab, one thing we did was really think about what are the challenges with doing this type of in situ microbiome engineering, right? So how can we change our kind of framework from engineering just a single E. coli species in pure culture to engineering a community full of non-model microbes? And what we thought about really was, what are some of the big challenges, technical challenges that stop that type of genetic engineering? And then how could you innovate new types of RNA technologies to kind of address those challenges? And today I'm going to just tell you about three short stories, kind of focusing on different challenges in this space. So the first challenge is we need to get a lot better about think about how to introduce DNA into communities of non-model microbes. And today I'll tell you about how we've kind of trying to solve this through creating synthetic genetic memory systems that allow us to, for the first time, autonomously record the movement of DNA in these communities. Secondly, once you have DNA delivered, for certain applications, it's going to be desirable to control that genetic information, right? So for example, you may want a genetic program to only turn on in a certain identity of microbes. Great example for why we want to do this is we were really interested in creating genetically encoded antimicrobials. And so today I'll tell you about our work trying to create RNA sensors that can sense host cell information and use that to control the activity of a genetic program. Finally, we're really interested in thinking about how to control cell function and phenotype, and this really boils down to creating synthetic RNA regulators of gene expression that we can use to turn on and off different phenotypes. Okay, so I'm going to tell you about kind of quick short stories from these three different spaces as time allows. And so the first thing is really thinking about this first challenge, right? So how can we get better at introducing DNA into communities of non-model microbes? And as you think about this problem, I'm sure many of you are thinking, okay, a good solution to this is probably we could exploit the natural mechanisms of horizontal gene transfer, right? So think transduction, conjugation, transformation. This is how microbes naturally exchange genetic information. And of course, we're very good at doing things like transformation on our model microbes like E. coli, right? Probably many people on this call have done a chemical or electrotransformation of things like E. coli. Now, while we're very good at doing that for our model microbes, our understanding of how HDT occurs in real communities and real environments is actually very, very, very limited. And so why this limited understanding? Well, it's really because we don't have good assays to measure gene transfer in communities. And so the most common way to do this is protein-based reporters. So think, fluorescent proteins or antibiotic resistance genes. And so these are very hard to apply in natural communities. Most natural communities are composed of microbes that we can't really culture, so that's very limiting. And of course, many of the communities that are interesting are residing in complex environments like soil, and so you can't really do things like fluorescent measurements readily in soil. And so we got really interested in trying to solve this in a highly collaborative project with two labs at Rice, the Silberg Lab and the Stadler Lab. And our big innovation here was to try to think about how we could use a synthetic RNA memory system to autonomously record when cells take up DNA in a community. Okay, so that's going to be the first story I'll tell you about today. What we'll do first of all is try to understand how the synthetic RNA memory system works. And so if you imagine I have a community of different microbes, and my goal is to understand which microbes in that community take up a particular DNA plasmid. Okay, well, how this memory system is going to work is we're going to first embed into this DNA that we're monitoring this blue DNA sequence, and this is going to serve as a barcode. Okay, so when a community member takes up this DNA, what happens? This DNA comes in, and this DNA barcode will get converted into an RNA barcode. Okay, now what we're going to do through a special biomolecular writer that I'll tell you about in a second is we're going to attach this RNA barcode onto a host RNA shown in red. And we're going to choose a host RNA that is unique to each individual community member, so that if I sequence it, I can tell exactly which community member it came from. Okay, so what I've done basically through this barcoding reaction is created a memory, right? Basically, that the cell took up the DNA. So how can we read out this memory? Well, we can do this through sequencing, of course, right? If I sequence the blue part of this barcoded RNA, that tells me which DNA entered the cell. And if I sequence this red part, if I've chosen a host RNA that is unique to each individual community member, well, then I can tell which cell this event occurred in. And so this we thought was a kind of new concept to the field. We called this technology RNA adjustable modification, or RAM. And if you're interested in learning more about it, the paper came out in Nature Biotechnology a couple of months ago. Okay, so this is a cool idea and could solve a lot of problems in the field. What do I need to do to get it working? Well, I really need two things. So the first thing I need is I need a host RNA that's going to serve basically as the my information storage location. And the second thing I need is I need a biomolecule that can write these RNA barcodes. So let's think about that first problem first and ask the question, okay, what biological molecules can write RNA barcodes? And so for this, we turn to a system that we work with a lot in my group, which are engineered trans-splicing ribozymes or catalytic RNAs. Okay, so we'll see these a couple of times. High level, how they work. Okay, so these are not RNA systems. These are ribozymes or catalytic RNAs. This particular form is a synthetic ribozyme, so it's been engineered to perform a splicing reaction in trans, so onto a separate RNA species. Okay, so how this works is we use a truncated form of this natural ribozyme. We amend onto an RNA guide, kind of like a CRISPR-Cas system, and we design this guide to be complementary to the host RNA that we're going to barcode. Now, when these two RNAs are present in the same cell, what this ribozyme or catalytic RNA is able to do is perform a splicing reaction, a trans-splicing reaction, where it joins this blue barcode onto a uracil in the host RNA to form this spliced RNA output, okay? Which in essence is our barcoding reaction. Okay, so these systems are really nice. You can change the guide, you can change the barcode. They're very modular. And in our lab, we do a lot of work to really understand the design rules of these systems. And so how we'll often do that, just to give you some insight, is we'll often use a fluorescent splicing assay. And so I won't go into too much detail here, but the general idea here is we're going to have a splicing reaction where the output is going to be a mRNA that encodes for a fluorescent protein GFP. And so this is really nice because what we can do is basically measure GFP as our proxy for splicing. And so just to give you a sense of how well these systems work, this is fluorescence data in E. coli cells where we've transformed different plasmids, okay? So cells is where we've transformed just an empty plasmid, so that's our autofluorescence, nice and low. Positive GFP is a positive GFP control. So this is a nice high on signal. And then in blue is one of these catalytic RNAs. And you can see compared to our positive control, we see about 25% splicing efficiency. That's pretty good, right? So we're pretty happy with, particularly for this type of application. So through a lot of hard work, we can create these kind of RNA writers, and we can really optimize their efficiency. And I'm not really showing you too much of that work, but you can find the details of how we did optimize them in the paper. Okay, so this is part of the problem solved. I have my writer, my RNA writer. The next part of the problem I need to solve is I need to find a good host RNA that I can barcode, right? Which is basically, in essence, this host RNA is going to serve as my storage location. Okay. So what RNA is a good storage location? Well, I really want two things from this storage location. So the first thing I want is that I want a host RNA that is going to be ubiquitous across diverse microbes, right? So regardless of who is taking up the DNA, I want this host RNA to be present, okay? But the second thing I want is I want this host RNA to also be unique. So if I sequence it, I can say, okay, yes, this is from this microbe and not this microbe. So it turns out the perfect RNA for this is actually 16S ribosomal RNA or rRNA. Okay, so maybe people are familiar with this, maybe not, but 16S rRNA is the rRNA that we most often use to taxonomically classify different microbes and their species. And the reason we use this particular RNA is it has this interesting kind of striation pattern to the gene. Okay, so what we find for this gene is we find highly conserved regions in gray that don't really change across the domain of life, like bacteria, flanked with these highly species variable levels shown in these colored bars. And so this is actually really perfect for our particular application because we can design one of these splicing ribozymes uses an RNA guide to target these highly conserved regions so that regardless of the microbe taking up this DNA plasmid, we can always get a barcoded product. And then when we sequence this barcoded product, well, as long as we go into enough sequence variable regions and colors, well, we can then figure out exactly which microbe this event occurred in. And so this is a cool idea. Would it work? So what we did to test that is design a bunch of these different catalytic RNAs to target different conserved sites. I'll just show you one design that we ended up settling on. So this is now qPCR data, so where we're quantifying the transcript copy numbers of E. coli cells that have a plasmid that has one of these memory systems and a plasmid that doesn't have. So minus RAM is just a regular plasmid. We shouldn't see any barcoding. And then plus RAM is where we transform cells with a plasmid with one of these memory devices. In gray, I'm showing the quantification of just native rRNA. So that's what, like kind of non-barcoded wild type rRNA. That's the same regardless of the plasmid being transformed. But now if we look at the barcoded rRNA, okay, we see a nice strong signal when we have a plasmid containing this memory device. So that's nice to see. Okay. So in the paper, we did lots of other analysis to show, the half-life of these barcoded signals and other things. I'm just going to go straight actually to applications of this to measure gene transfer in a complex community. Okay, so the experiment we settled on, one of the experiments, was to ask the question, well, could we use this memory system to measure the conjugation of a plasmid from an E. coli donor into a complex community, in this case, a wastewater community? So the idea was we could take a wastewater community. We actually went to our local wastewater treatment facility down here. This is Rice University over here. Took a community from this wastewater treatment facility, took it back to the lab, added in some E. coli, let it incubate for 24 hours, and then asked the question, okay, do we see conjugation? The type of sequencing analysis we have to do for that particular experiment is first we do native 16S rRNA sequencing. That tells us who is in the community. And then we do this special barcoded 16S rRNA, which tells us who took up the plasmid. Okay. So the data we get from these experiments is actually quite complex. I'll try to break it down a little bit. So what I'm showing you here is a wheel of all of the microbes we find in this community. This is a complex community. We find about 300 different species. Okay, they're ordered based upon how related or unrelated they are. And what I'm showing you initially in these orange bars, these are the microbes in that community that we detected taking up the DNA plasmid. Okay. So initially, something very shocking from this experiment, which is actually of these kind of 300 communities, 300 members of this community, around half of this community could actually take up the DNA plasmid from this one E. coli donor. Okay, and this is actually shocking because this is way more uptake than we were anticipating based upon the literature. Now, you can get lots of other information from this experiment. You can get discrete values of the individual barcoded and native signals. Other things you can learn is who in the community is taking up this DNA. And so here what I have are basically the different orders that we find in this community. And on the right here is a bar graph where I've plotted the fraction of different community members from each of the orders. And so you can see, well, the microbes that are taking up this plasmid a lot are things like Aeromonas, Enterobacterials, Pseudomonas, but in general, actually quite a diverse set of microbes are able to take up this plasmid. And again, that's kind of shocking because the literature was such actually that a narrow range of microbes should take up this plasmid. So this is a really nice technology. You can do other things with it. So you can start to learn the design rules. So for example, ask the question, which plasmid would a community take up more efficiently? And so I'll quickly show this experiment. In this experiment, what we did was we take two different plasmids that vary in their origin of replication. Okay. We amended onto each of these different memory devices. In this case, we actually included a unique barcode so that we could tell exactly which plasmid signals were arising from. We put them into E. coli donors, and again, we added them into this wastewater community. Again, you get a lot of data out of it. I won't go through the details, but high level, again, we see a lot of uptake of these two different plasmids. Around 200 take up this one type of origin, PBR1. About similar number take up this other plasmid, P. coli 1. Many are able to take up both, but we also do find many situations where a microbe will take up one plasmid, but not another plasmid. Okay, so we think this RAM technology is a very powerful technology for microbiome engineering. It's going to really allow us to understand, the design rules of how to get DNA into different types of communities, as well as understand the fundamental processes of gene transfer. Kind of reflecting on where this type of technology I think could be very useful for synthetic cell community. I think something I think about a lot is the risk of gene transfer from engineered microbes or synthetic cells to natural communities. Okay, and I think technologies like RAM are very useful technology to understand what that risk truly is, right? So in ongoing work, we're taking, for example, engineered microbes and understanding what the rates of gene transfer are from that engineered microbe to natural community members in environments like soil. I think similar types of experiments are going to be valuable for synthetic cells. I think also for biosensing applications, it's very powerful. I think there's a lot of interest right now in the field of synthetic biology to engineer microbes as well as synthetic cells to uptake eDNA as a form of biosensing. So again, I think RAM is a very powerful technology to start to understand those rates. Okay, so that's kind of this first project area that I wanted to talk about. The second project area is think about control. Okay, so let's say I get to the point where I can deliver, highly efficiently DNA to many community members. For many applications, it's going to be valuable to have some sort of spatiotemporal control, where, for example, a genetic program might be on in one microbe, but off in another microbe. And so this is a space that we've been very interested in for a long time. And really how we thought about trying to tackle this is create RNA sensors, right? So our big idea in this space is to think about, okay, could we use native messenger RNA signatures in cells to control the activity of genetic programs, right? And so why we think this is kind of an interesting idea is, within a cell, all of those transcripts, those kind of RNA signatures, they tell us useful information about that cell, right? They can tell us what species it is. They can tell us what phenotype is being displayed. They can tell us, is the cell stressed? Is the cell not stressed? And so if we had a way to sense that information, and then convert that into orthogonal protein outputs, well, that could be very valuable, we think, for many different types of application, whether that's biosensing, whether that's trying to create genetically encoded antimicrobials, whether that's for metabolic engineering. So today I'll just tell you about one of our efforts in this space, which was the thesis work of a really talented graduate student, Dr. Lauren Gamble, that published a few years ago in Nature Communications. Okay, so what Lauren wanted to do was create this kind of RNA transduction platform. And a particular platform they created, we ended up calling Render, which was this ribozyme-enabled detector of RNA. So again, this is actually based upon an engineered splicing ribozyme. What I'm showing you here on the left is actually the natural splicing ribozyme reaction. And so these splicing ribozymes, they're naturally found embedded in between genes. So this is black sequence, which encodes for the ribozyme, is embedded between these two gene fragments in blue. And naturally what happens is this whole thing gets transcribed as a single RNA species. And what happens when this is allowed to fold is this ribozyme performs a splicing reaction where it basically joins this right exon onto this left exon at a uracil to form the spliced RNA output that can then encode for a protein. Okay, so Lauren's idea was to take this and engineer it so that it was dependent upon an RNA input, basically, so it could do a sense and reaction. Her big idea in this space was to take this ribozyme, synthetically split it into two non-functional halves, right? So where we basically destabilize the structure so it can't come back together, and then amend onto those halves RNA guides that are designed to detect an RNA input. So the idea will be, without this RNA input, these two halves can't come together to form the tertiary structure and this splicing reaction. However, when this RNA input is present, well, that can bring these two halves together, stabilize them for long enough to produce a spliced RNA output, which can encode for a protein output. Okay, so on paper, again, this seems like quite a good idea. These inputs could be quite designable by changing the RNA guide, and we know from other work that you can easily change these exons. So in theory, you can connect any input to any output. Okay, so the first thing we had to do in this project that turned out to be a little tricky was figure out where to split this splicing ribosome. Okay, so the structure of this ribozyme is shown here on the slide, and as you can see, it's quite a complex, secondary and tertiary structure. It's around 400 nucleotides long, and so the first thing we need to do is figure out where is best to split the ribozyme in this structure. And what we're looking for in a split site is basically to find a site where, when we don't have the RNA input present, this thing is destabilized, so the two halves can't come together. However, when we do have the RNA input present, these halves are able to come together and get complemented. Right? So in other words, we need a split site that has a nice low off state of splicing when the RNA input is absent, and then a nice high level of splicing when this RNA input is present. So to do this, what Lauren, alongside another very talented graduate student, August Stabus, did was a high throughput selection. So they built every possible split site, they coupled it to a GFP output, and then they performed a round of fluorescent activated cell sorting to basically find sites that had low fluorescence in the absence of an RNA input and then high fluorescence when that input was present. The data From that experiment is shown here on the right. This is called a relative enrichment plot. How you're going to read this data is as follows. So the x-axis represents different split sites along the ribozyme. And then the score here is the level of enrichment we see through the fluorescence-activated cell sorting. Okay, so high level of the sites along in the bottom box are sites that were de-enriched during the selection. So these are sites that are not good. So either they have kind of leaky off states, or they maybe have low on states. However, on the top are the sites that are enriched. And so these are basically the functional sites where we have a really nice detection reaction occurring. You can take this data and overlay it on the structure of the ribozyme, and then you can actually learn some interesting design rules. For example, we see regions like this P9 region to be highly enriched in split sites, right? And there's some structural and sequence reasons why this is interesting. This in the three-dimensional structure of the ribosome actually wraps around the structure of the ribozyme. And so that kind of makes sense why that's a good place for split sites. All right, so what we did next was basically take a series of these enriched split sites and then individually characterize those variants for their ability to perform an RNA detection reaction. And so the data I'm showing you here is where we're using a fluorescence output in an E. coli cell where we're detecting an RNA input. Okay, so gray is the off state, blue is when the on state, when you have the RNA input present. What we see, well, across all the designs, we actually see a nice low off state. Basically indistinguishable fluorescence compared to blank E. coli cells. However, in blue, what we see, well, nice high on states, right? Particularly for some of the more optimal split sites. So this is really nice. This shows us that we can kind of create these RNA sensors with this nice high dynamic range detection that we can start to use towards application. Now, again, we did lots of other experiments in the paper that you can read about if you're interested. For example, showing that we can redesign the input, redesign the output. What I wanted to do is just end on this part of the story by just showing you one application of these sensors. So in this case, what I'm going to show you is Lauren's efforts to create a genetic program that would be able to sense host cell phenotype. Okay. In particular, what she was interested in sensing was a cell resistant to an antibiotic, in this case, kanamycin. So what Lauren did was design a sensor that would sense the kanamycin resistance transcript, and in response, produce a protein output that encoded for an enzyme called FMO that produces the pigment indigo. Okay, so one can imagine why this could be useful, right? It'd be a way to create sort of a biosensor that would detect if a cell's resistant. You could easily replace this enzyme with a death gene so that it would kill cells that were resistant, for example. So Lauren took this design, she transformed it into E. coli. Here's the results that we saw. So blank is just when you transform E. coli with a blank plasmid. So we see no production of indigo. Minus CANAR is when we transform it into a plasmid that isn't resistant, and then plus CANAR is when we transform the sensor into a plasmid that is resistant. And so what you see, again, what we saw before, nice low off states with this very, very strong production when we do have this input present. Okay, so that's the second kind of story I wanted to tell you. This is a space we're very interested in, generally for genetically encoded antimicrobials or other applications. Now, what I wanted to end on was really this kind of third project area, which is our work trying to create synthetic regulators of gene expression. Okay, so like many people in synthetic biology, and I'm sure also in synthetic cell research, we're very interested in thinking about how to create gene regulatory networks, right? So gene regulatory networks are, of course, these networks of gene regulators that cells have naturally evolved to process information, right? So to convert lots of different input signals into cell decisions, right? Deciding which cell phenotypes to turn on and which cell phenotypes to turn off. And so like many, we've been interested in thinking about, okay, can we build the kind of basic parts of the networks, activators and repressors? Can we engineer them to have desired characteristics like orthogonality? And then from these start to build, more complex kind of genetic network motifs, one-to-many, autoregulation, signal integration, etc. Of course, the unique thing about the work that we do is we're very interested in trying to build these networks, not out of proteins, but out of RNA. Okay. So one reason we really like RNA is this idea that, well, if you create transcriptional RNA regulators, there's potential to build networks that are composed entirely of RNA. So in other words, they're RNA-only networks. The input can be an RNA, and the output can also be an RNA. And there's some specific advantages to do this. First, in terms of complexity, you're just using RNA, so there's no proteins. In theory, and we have some work from Julius's lab showing this, signals are propagated faster if it's an RNA-only network as opposed to if it's an RNA protein network, and other potential advantages. Okay. So I've been in this space for a long time. One of the major contributions as a postdoc was to try to create transcriptional activators of gene expression that were RNA-based. And so we created, in Julius's lab, a technology called STARS, small transcription activating RNAs. And so these are a very synthetic regulatory mechanism. It doesn't exist in nature. But as the name suggests, it's a mechanism where we turn on gene expression using an RNA. So how this works, well, let's say there's a gene I want to regulate. What I do is put upstream of that an RNA called the target RNA. Now, what's interesting about the target RNA is when this is able to be produced and fold, by default, it folds into a transcriptional terminator hairpin that turns off transcription of the downstream gene. So when you put this target RNA upstream a gene, by default, it's going to turn that gene off. So you don't get transcription of that downstream gene. But what you can do to turn on the expression of this gene is express one of these short RNAs that's able to bind to the terminator, prevent its formation, and allow transcription of your downstream gene. And if you're interested, you can read about the papers describing the discovery of this mechanism. So this is a very synthetic mechanism. It works very well. This is now just characterization in E. coli. So I'm showing you now just some flow cytometry data where we have the output in fluorescent proteins versus the distribution in the population. And what you see if you compare the on-state in gray to the off-state in gray to the on-state in green, nice high-fold activation. Okay. Now, while this work was important and did push forward the field of RNA synthetic biology, one of the limitations was that we only did this in E. coli, right? So we only did this in E. coli, and so one thing a graduate student, former graduate student Bayang Liu, got very interested in was to understand, okay, well, could STARS represent a really powerful regulated gene expression that will function across diverse microbes? And so the work I'm going to tell you about today was, again, a Nature Communications publication from a couple of years ago, which you can read about if you're interested. Okay, so what Bayern was interested in doing was taking the STAR system and asking the question, could this function across diverse microbes? And so when he put this system into a bunch of different diverse gram-negative microbes, indeed, he did show that it was functional. So the off state is in gray, the on state is in blue. What you see is across all these very diverse species, we see nice high-fold activation. Ban also showed that you could actually use phage polymerases and the system would still work. And so this was really nice because it showed that these STAR regulators, as well as the circuits you build from them, are going to function across a diverse range of microbes. Now, another key discovery and insight described in this paper was this kind of new idea about how you could tune the gains, this relationship between input and output of these STAR systems. So Bang in this paper described a really novel idea that's very creative and very synthetic, which is the idea of RNA regulatory arrays. Okay, so let's say there's a particular STAR that I want to tune its input-output relationship of. Well, the idea of a regulatory array is this. Let's say there's a promoter that's giving me a transcriptional input. What I can do in a regulatory array is basically tune the copy number of different stars, how many stars I have encoded in a single transcript. Okay, so I could change this from 1 to 2 to 3 to 4. And as I change the number of inputs, that should, in theory, change how much output I get. In other words, this can serve as an amplifier. And so in theory, how this works, well, you take a single promoter, you could then put one of these regulatory arrays downstream of it. This would have a series of regulators encoded in it, and then this would all be produced as a single transcript. Now, to separate out the individual regulators, what we envisioned was encoding different insulator sequences, and so these are going to be things like self-cleaving ribozymes or CSY4 hairpins that would then separate this single transcript into individual functional regulators. Okay, so really cool idea. How do you get something like this to work? Well, I won't get into details. We did a lot of RNA engineering to figure out how to get good insulation to be able to cleave these different transcripts. Instead, I'll just go to show you some characterization in bacteria. Okay, so what I'm showing you here is a graph where we have plotted the input, so promoter strength, into different regulatory arrays that vary in their copy number of stars from 1 to 8 versus the output signal. And so what you can see is as you vary the copy number, you get this nice predictable increase in the output. You can see this really nicely actually if you just focus on, for example, the characteristics when you have the strongest input present. And so again, what I'm showing you here is output versus copy number. And what you see is this very nice predictable relationship. Okay. And so this is really shows the benefit of this system, right? Which is that through this kind of array approach, you can get this very predictable tuning of your regulator's performance. So this is characterization in E. coli. You can take this and do this in a bunch of different species, as we did. I'm just going to show you one species. So this is now a different microbe. This is Shewanella. This is an electroactive microbe, so people are very interested in this for bioelectronics. And again, when you compare the characterization data, it works, and it's very similar to what we see in E. coli. It's kind of very predictable tuning of output. Okay. Final thing I'll show you today is asking the question, can we use this RNA array approach to tune and implement genetic circuits? Okay. So like many people, we're interested in different types of motifs. Particularly, we're interested these days in cascades and multiplexed outputs, in part because this is, of course, how you would start to think about building neural networks inside of living cells. And so, we were really interested to say, okay, well, could we use this array approach to implement and tune these different circuits and also implement them across different species? So I just focus on this bottom motif for now and show you how we can use arrays to tune the performance of this. Okay. So I'm showing you here now is a high-level schematic of a multiplex output. Basically, we take one input and control two different outputs, output 1 and output 2. How that looks in terms of genetic circuits is shown on the bottom here. So we have, for our input, a single promoter. This will drive the transcription of first this one star over here in orange. That's going to control one output. So GFP, we'll call it output 1. And then another output, which is another star. This is going to control a different output, RFP, we'll call that output 2. And so how you can use arrays in this situation is basically you can individually tune the copy number of this star 10 and this star 50, right? So I can double star 10 to double this output 1, or I could double star 50 to double the output 2. Okay, so does this work? Well, let's first look at E. coli. So I'm showing you here is basically characterization of GFP and RFP with different array designs. And so first look at GFP. What we see here is when we double the star copy number of the star control in GFP, we see a nice doubling basically of the GFP output. However, when I change star 50 copy number, which remember controls RFP, I don't see any difference in GFP. Counter to that, if you look at RFP, you see the opposite. Change in star 10 copy number doesn't change the RFP copy number. However, change in star 50 does change the amount of RFP we're able to produce, right? And so this is really nice. You can think of this another way in terms of weighting, right? Basically, what we're doing here is weighting the different outputs, very similar to how you do in neural networks. Really nice thing again is you can take this design in E. coli, you can implement it in a very unrelated microbe, Shewanella, and it basically performs the same. And if you're interested, we also did in the paper the same thing for cascades. Okay. So quickly to end on, I just wanted to highlight some other work in this space because I think this does connect well to synthetic cell research. Another paper that Bayang published in collaboration with the Lisa Franco lab was a really interesting circuit design that enhances modularity. And so this we call the RNA compensator. It can basically compensate for what's called load. Okay, so it's a way to achieve consistent signal when load varies in a circle. Another paper we're hoping will be out soon is the synthetic origins of replication. And so in this paper, what we did was we took synthetic RNA regulators and used them to create origins of replication that were more tunable, more orthogonal, and more stable and compatible with other plasmids. We have a submission on bioRxiv about this if people are interested in, and we hope to have the paper out soon in the coming months. Okay, so just to end on the kind of relevance of this last part of my talk to synthetic cell research, well, of course, when we look at naturally evolved microbes, they're mostly reliant upon proteins for these regulatory networks. And I think, there's many potential advantages to switching to an RNA-only network of gene expression. In theory, it's more faster, it's designable, and potentially more orthogonal. Other reasons I think this is relevant for synthetic cell research, one place we do see naturally evolved cells use RNA regulators is in responses like stress response. Most of the stress response systems of a cell actually use small RNA regulators. And so I think, when you think about synthetic cells, there's probably advantages to use an RNA for certain types of responses you're trying to program. With that, I'll just thank everyone in my lab who did the hard work, particularly Bayang and Lauren Gamble, whose work I showed today, our collaborators as well as our funding agencies. And with that, I'm happy to take any questions.
Thank you so much. I really enjoyed it, especially because I deal with a lot of those problems myself. There is a lot of questions in chat. Can you see
Okay,
chat?
I can see chat. So should I read the question out and then?
Yes, please.
Okay, perfect. Let me go through that. Okay. Okay, so I'm seeing, yes. First question is, so the first question is from Keaton. It's what strategy, like guide RNA chain length, was implied to avoid the cross interaction with truncated ribozymes? Yeah, great question. So I think the question gets at these transplicing ribozyme systems where the interaction is governed by an RNA guide versus a target RNA that you're binding. In general, the length of those, you can do however long or short you want. Optimal is around 25 to 50. And in terms of avoiding cross reactions, what we did there was more computational. So when we designed the RAM system to target 16S ribosome RNA, we just made sure that those sites that we're targeting were not found in other parts of the genome. And so kind of more bioinformatic analysis. We did do some characterization. For example, we measured the burden on cells when they express these systems. We sequence what RNAs they're barcoding. And in general, they're pretty specific and don't add too much burden. So we don't think they're targeting a lot of other things in the cell. Okay. Second question from Drew. Yeah, this is an awesome question. Okay, so the question, maybe to rephrase it. So, I showed, maybe I'll pull up my slides. Okay. Yeah. So I showed the work where we are, yeah, perfect. Barcoding, maybe I'll go to this slide. Yeah. Barcoding 16S to create a memory of gene transfer, right? So the question is, how do you avoid degradation, right? Won't after some time this species just get degraded and then you've lost your memory? That's indeed very true. So this memory system that we created is not necessarily permanent memory like you associate, let's say, with DNA recorders. This is more temporal memory, right? So if for whatever reason, let's say a cell takes up a plasmid and then it loses a plasmid, it would lose a signal over time. Half-life of this RNA is not particularly long. In pure culture, growing under ideal conditions is probably about 20 minutes. Okay, so that you can see as a limitation, or you can see it as an advantage. It just really depends on the question you're asking. So if you care about asking the question, okay, in the lifespan of this microbe, has it ever taken up DNA? Not whether it has it currently, but has it ever taken up? There you really want DNA recorders. Now, if your question is more, does it currently have DNA or has it, acquired DNA then lost it? That's where you really actually want temporal memory, right? You want to know, does it have it in the instance? So again, I think we see RAM as complementary to some of the DNA recording technologies, particularly if you're interested in dynamics, right? If I want to understand the dynamics of gene transfer. Yeah, great question. Okay. Yeah, great question. So the second question from Kayla is, in a situation where I want to, let's say, have two plasmids I'm trying to record the uptake of, what happens if a single cell takes up both of those plasmids, so it has barcode 1 and barcode 2, like how are you going to get a signal? So that's a really fantastic question. Yeah, so in these kind of situations where we have, I don't think I have a good slide, but two barcoding systems. Okay. So that's actually not a huge issue for us. So when we do this barcoding reaction, we actually only barcode a very, very tiny amount of the host cell 16S ribosomal RNA. So we target probably, 0.1, maybe even 0.01% of that RNA. And so, why we don't barcode 100%? well, likely cells would be very unhappy with this if we started barcoding a lot of the RNA. So we actually only barcode a very, very small fraction, enough so we can sequence it and detect it, that's fine, but not enough to be able to impose a lot of burden on the cells. And so in this scenario where one cell takes up two, there's plenty of native or unbarcoded RNA for both systems to work. So that's a really great question. Okay. The next question, yeah, great question. How long around does a RAM modification persist? So, after a plasmid, a cell would drop a plasmid, let's say, how long would you expect the signal to last? So yeah, great question. Generally about 20 minutes or so is how long the signal would last. We have a follow-up paper coming out where we have really redesigned the system a lot, but one of the things we tried to redesign was stability. So we tried to make the signal much more stable. And why we did that was basically just to reduce the half-life so we get an overall increase in the signal that can help with some of the downstream processing where we're amplifying it and sequencing it. Would RAM like tech work in cell-free? Is the next question. It's an awesome question. Yeah, we actually are trying right now to do work with the ribozyme system in cell-free. So I think the initial results suggest the ribozyme does work. So I think a technology like that would definitely work. Where you've got to be careful, I think, and we've seen this when we've tried to use RNA regulators in cell-free. You really want to control things like magnesium and other things like that that can really affect function. So if you're interested in that, if you look at some of Julius Lux's publications, we had some papers when I was working with him where we really showed how you could use cell-free systems to characterize RNA regulators. And in that work, you'll see some of the kind of optimizations we did of the cell-free systems. And magnesium is one of the things you need to be a little bit careful of, but in general, RNA systems work pretty well. Okay. Next question is about, how sensitive is render to mutations in the RNA input? And can you apply it for the detection of mutations? Okay, yeah, that's a really awesome question. So we didn't directly characterize this, but we do think the system is quite tolerant of mutations. And so, again, you can sense different lengths of RNA. So, you sense short, like 50 base pairs. You can sense long, like 1,000 base pairs. The longer you are, the more tolerant you are. But in general, they're pretty tolerant. So you can do mismatches and the systems will still function up until a point. The second question is actually really awesome. Like, can you detect a specific mutation? We've never tried that. I think in theory you can. How I always imagined that working would be, I could go to the slide for this. Yeah, is something like this. So, people have shown that you can design these interactions to be really on the edge of stability, right? Where a single mutation drops you off the cliff, so you're destabilized. Whereas if you don't have that mutation, you have just enough energy to hold the reaction together. So there's some really awesome work from Alex Green's lab doing this with total switches. And so I think if you have a system like that, in theory, you could get to the point where you detect a mutation, but we haven't done that in my lab as of yet. Okay, I think that's the only question I see from the chat right now, but I'm happy to take more of people unmute themselves or whatever, Kate, whatever people normally do, I'm happy to do.
I I have a question, and
Yeah.
if anyone else has questions, please chime in. But my question is, it might be naive, but how many barcodes can you do simultaneously in the same cell?
Yeah, that's an awesome question. We've never pushed it. I think it is massively multiplexable. I don't think there's any limitations, right? So the barcode itself, so it's a great question. Let me get to that section. Yeah. So if you read the details of the paper, that was the barcode we actually used for this paper here is just a fragment of GFP. So it's a barcode in the sense of it's a sequence we know the identity of. In the follow-up work, we've been much more deliberate about designing barcodes. And so we have a really nice modular strategy now where you can change the barcode for any sequence. And so I think it's basically limitless the number of barcodes you can make in a function. How many would be tolerated in a cell? It's a good question. I do think there might be some challenges with recombination and stuff if you had a lot of these systems, but I think they're going to be more. I wouldn't be shocked if you can easily get to 10 without problems and probably even more. Yeah.
That's awesome. That's really exciting. I'm really looking forward to using RNA regulation. The reason I ask how many barcodes is because I want to do what you suggested is going to be possible, which is use RNA regulation instead of peptide-based systems for controlling a complex synthetic cell metabolism.
Yeah. Yeah. No, I think that's a dream I've always had. I've been more on the cell focus, but I think the dream is the core, which is like, can we get to, these complex, this is just 20% of the regulatory network from E. coli for proteins. How can we do that with RNA? And I think there's really great synthetic RNA regulated systems where now we can really see what's possible. And I think doing it in a synthetic cell is the way to go. Yeah.
Oh, yes. All right. Thank you so much. If there are no more questions, I want to say thank you, James, again.
Great. Well, thank you so much, everyone, for listening. And Kate, thank you so much for the invitation. And
Thank
yeah, if people
you.
have any questions or interest, just ping me and send me an email. You can find me online, and I'm happy to respond or assist in whatever way I can.
And have a great rest of your week.
Great. Thanks.
Bye. Bye.